A very good evening and namaskar to all. HBNI Women's Cell is extremely glad to arrange a webinar today. Today is a Makar Sankranti. I wish everybody a very happy Makar Sankranti. To join us, we are fortunate to have amongst us Professor Vidita Vaidya, Department of Biological Sciences of TFR, Mumbai, who joined us online. Our heartiest welcome to you, ma'am. On behalf of HBNI Women's Cell, I'm welcoming Vice Chancellor, Dean, members of Women's Cell, faculty members of CI, OCC, student, my colleagues from Central Office and HRDD who all joined online. Now, I request Dr. Uma Shankari, Chairperson of HBNI Women's Cell, to introduce the speaker. Dr. Uma Shankari. Sir, we are extremely sorry for the delay. Dr. Uma Shankari has not joined yet, sir. We will just wait for a few minutes. There was some technical issues. Uma Shankari madam को कॉल करें 20483. Sir, I request Professor Vasudeva Rao, Vice Chancellor, to just welcome our speaker, sir. Sir, किधर गए?
Very good afternoon and a warm welcome to Dr. Vaidya for joining us today to educate us on this very intriguing topic. I wish you all a happy Makara Sankranti. So when I received the announcement of the webinar, the title was really French and Greek to me because I am a chemist by profession and this topic was very, very peculiar. So I thought I should do little bit research. So I went to the homepage of Dr. Vaidya and found that this work relates to molecular psychiatry. And that made it even more intriguing for me because I have heard of molecular spectroscopy and several other things, but not molecular psychiatry. And I also read that it is slightly different from biological psychiatry. Well, this made it very, very interesting for me. And I decided that even though I am at home with COVID, I should certainly join this meeting and listen to her and get some idea of what this topic is about. But one thing I have learned is that this is a subject of very great interest and importance today because I could see that even Nature runs a journal on molecular psychiatry and with a very, very high impact factor of maybe 15 or 20. Maybe Dr. Vidita Vaidya has even published there, I do not know. I was trying to think how old is this subject and tried to read some articles about this in the journals. Then I came across a very interesting article which gave me about the origins of this term, molecular psychiatry. And I found an article by Eric Nestler in the Journal of Molecular Psychiatry in the year 2013, which discusses the origins of molecular psychiatry. And it is in that article that I learned that this term was itself coined as late as 1987. It's around, that means subject is, in this form, the subject is maybe around 35 years old. It was very interesting that the way it was coined at that time, I was told that just like me, several other colleagues at that point of time were very much intrigued with the title. Dr. Nestler writes that one day in 1987, I was setting up my laboratories in the Connecticut Mental Health Center, and I decided to name our laboratory corridor as the Laboratory of Molecular Psychiatry. and he says immediately that several colleagues of his had very different types of reactions to this, and to the extent that one of the friends posted a sign on his laboratory and called it as laboratory of molecular fear, and another called it as no, laboratory of molecular psychoanalysis, just to compete with him. So they were trying to make light of the issue, but as the article goes, it talks about how important it has become and how its goals are so important to society today, to understand how molecules can influence the thinking process and what is the connectivity. So I think it's a very important subject of the day, and it is very nice of Dr. Vaidya to join us, to enlighten us on the subject. I welcome her to this program, and it is for Dr. Bharati to decide whether we wait for Uma Shankari or we proceed. You are muted. We can't hear Bharti.
Yeah. Sir, we will start. We will not wait for Uma Shankari because there is some technical issues is on, sir. So I request Madam to start with her lecture.
Yeah. Thank you so much.
Okay,
that was a very kind introduction to the field of molecular psychiatry. The lab that you talked about and that corridor is exactly where I did my PhD in the 90s. So Eric and Ron were my thesis advisors. And so I was in that very same division of molecular psychiatry very soon after I started my PhD in 92. So soon after that term was coined, I showed up there to do my PhD. And so that's probably the genesis of how I got introduced to the field. Let me just share my slides and please let me know if you can see them. Are they visible? Are my slides visible?
They are coming on. Yes, it is visible.
Okay, great. So what I want to do today is talk to you about serotonergic psychedelics. And while the term molecular psychiatry is relatively recent, the idea that we use molecules to influence our mood is a very old idea. It's an idea that has been around for centuries and centuries, and you will see what I mean when I say that. Before I start, I'd just like to, first of all, let me just wish everyone happy Bihu, happy Uttarayan, happy Makar Sankranti, happy Pongal. This is a day of multiple celebrations across festival season and harvest season across the entire country. I also wish you well and hope you have a speedy recovery from COVID, sir. And yeah, hoping that the country stays stable through this third wave at the moment in time. Thank you very much for joining in to listen to this talk on this festival day. I'll just tell you a little bit about what my lab does, and then I'm going to introduce you to this topic of serotonergic psychedelics. So my lab is interested in two central questions. The first being an attempt to understand how early life experience, in particular early stressful experiences and the neurotransmitter serotonin, how do they modulate circuits in the brain that regulate emotional behavior? And we know this from psychology that early life experience can really change behavior throughout life. We're interested in asking how does it do this? What actually changes within the nervous system to mediate very long-lasting changes in behavior? And the other half of my lab is interested in the molecular pathways and the molecular mechanisms through which mood, anxiety, despair, emotional states in the brain are actually regulated, the receptor composition that is regulated, and the receptor pathways that are recruited to eventually bring about changes in mood. Okay, so let's go back to a history of psychedelics. And for this, we have to actually go back quite a way back in time, because perhaps the earliest idea that people took compounds to influence their mood is old. It's been, I mean, it's described extensively in Ayurveda, where there are compounds that are extracted from plants that can influence mood states. Much the same way all over the world, where variety of people were trying out different plant-derived molecules. There were certain plant molecules that were found to have very profound effects on mood. And perhaps the earliest examples of this is this particular mushroom, which has a molecule called psilocybin. Psilocybin is a constituent of mushrooms and can have a profound effect on the brain. And the earliest descriptions that it was actually being used are as far back as 4000 BC, because cave paintings actually have demonstrated illustrations of the psilocybin mushrooms as components of the cave paintings. And so it was used across the Americas in 3780 BC. So seriously, a very long time back. At that point in time, there's evidence that peyote is the active ingredient of this particular cactus. That was also used in indigenous ceremonies. And so across ancient times, it was used by a variety of people in different ways. There is this particular kind of tea that is brewed in South America. It's called the ayahuasca tea, which is a component derived from a couple of different plants. And this brewed tea is given by the shamans, who are the witch doctors, who actually use this as a way to connect with higher states, in a sense. And so the idea of, I mean, molecular psychiatry, the term may have been coined in the 1980s, but the idea that molecules can influence our behavioral states and our brain states is a very old one. And obviously humans have for a long time tried a variety of substances that can alter states in the brain. So this was used quite extensively, and sometime around 1500 AD, the Catholics actually referred to peyote as witchcraft, because when people consume peyote, it is associated with completely altered brain states. And because of that, you may hallucinate, you may have a variety of altered sort of consciousness states, which can result in a completely altered perceptual reality of the world. And because of that, it was considered to be witchcraft in a sense. And then, it was still being used, it was still being actively used. And these are the different compounds that we talk about. On the left-hand side in the yellow, ayahuasca is the tea that I described, and the core psychoactive molecule in that is dimethyltryptamine. Dimethyltryptamine here, which is a natural substance found extensively in many different plants, animals, even in humans. Peyote, which was the active substance of the cactus that I showed you. Ibogaine, which is derived from the iboga plant. Psilocybin, which is derived from more than 100 species of different mushrooms it can be seen in. And these are the synthesized molecules on the right-hand side. So lysergic acid diethylamide, LSD, which is a drug that came into our awareness in the 50s and 60s quite extensively. MDMA, which is again another synthetic drug. It's a methamphetamine. It's a synthetic drug which is also known as ecstasy. And ketamine, which is a synthetic drug which was used as an anesthetic, is also used as, it's a drug that is also a drug of abuse, but is used for procedural sedation as well. So these are the molecules that one is talking about when one calls them serotonergic psychedelics. And yet when you think about it, if more than about 6,000 years ago, this was actually being used, what has happened in the last 30, 40 years that resulted in this complete stigma around psychedelics, and where did that emerge from? Okay. So in 1897, let's look at the history of this again a little bit more. So in 1897, Arthur Heffler isolated mescaline, and mescaline is the active component of a cactus. Mescaline is again another serotonergic psychedelic. And there were attempts to isolate ibogaine, which came from the iboga plant, and all of this was happening. MDMA was created as a byproduct in 1912. But perhaps the big event that happened in this entire field of serotonergic psychedelics was the synthesis of lysergic acid diethylamide, LSD, which was synthesized by Albert Hofmann of Hoffmann-La Roche fame in 1938. And that singular event, he consumed a little bit by mistake. And when he consumed it, he had an utterly altered hallucinatory experience, because actually he thought he had consumed only a small amount, but it was quite a large amount, because LSD is extremely potent, and very, very minuscule quantities can produce very strong experiences of hallucinations and altered perceptual reality. So this happened in this period of time. And soon after is the discovery of serotonin as a neurotransmitter, the idea that these compounds are actually working in the brain on serotonin receptors came soon after that. And this led to an entire revolution that opened up the idea that serotonin as a neurotransmitter may be profoundly capable of influencing mood. Albert Hofmann also discovered psilocybin in 1958. Ketamine was synthesized, and all of this is going on. Now, remember, this is the period in which there was a strong hippie culture. This is also Vietnam War time. There is a lot of abuse of a variety of drugs happening in this period of time. And then there is a very strong lockdown on all drugs of potential abuse, including the psychedelics. So here are the molecules. Here's serotonin, which is the normal neurotransmitter that your body makes. Here is LSD, which is synthesized, the compound that Albert Hofmann synthesized. This is a molecule that mimics the effects of serotonin on specific serotonin receptors, one of which is this particular receptor called the serotonin 2A receptor. Here's mescaline and psilocybin. Mescaline, which comes from the peyote cactus, psilocybin from mushroom. DOI, which is a synthetic compound, which I work on quite extensively in our lab. And that's the compound that our lab is interested in. I'll tell you about that. Dimethyltryptamine, which is the active component of the ayahuasca tea. So now what's happening, right? Now, if you think about this period of time and just look at what happened like I told you, indigenous use of psychedelic drugs before any scientific discovery goes back thousands of years. It has been used long before anybody attempted to study it extensively in a scientific manner, but empirically it was used quite extensively, and it was extensively used by a lot of different people. Mescaline, LSD come in this period about more than like close to almost 100 years ago. And then in 1970, US passes this Controlled Substances Act, which shuts down all real attempt to look at this. In 1988, there's an identification that this compound LSD, which was a synthesized compound, works on the 5-HT2 receptor. And then slowly, there are a variety of studies that start happening. If you just want to look at the PubMed citations, okay, so suppose we say, let's look at how much has this work been cited, work on LSD, work on these different compounds. Here is work on dimethyltryptamine. There was a window in which it was extensively studied, then it died down, and then again, there's a resurgence. Ayahuasca was not originally studied, but there's a real emerging interest in this molecule. Here is psilocybin, again, an emerging interest. Here is LSD, big interest, shuts down and opening of an interest. These are compounds that have been synthesized more recently. These are all synthetic drugs that work at the 5-HT2A receptor in particular. And so they have been used more extensively in recent times. But there's a re-emergence of interest. And in a sense, what we're looking at is the beginning of something that's called the ongoing psychedelic revolution. This, in a sense, one can think about commencing from 2017. And this is very nicely illustrated in these diagrams that I've taken from a very interesting review that I cited a couple of slides ago. The resurgence of interest came from the fact that the Federal Drug Administration in the United States granted MDMA, which is a psychedelic, but is the amphetamine class, and it belongs to it's not a classical serotonergic psychedelic, certainly, but it's ecstasy. It is a drug of abuse. But this drug, when used in psychotherapy, along with psychotherapy use associated with it, it causes breakthrough therapy results, especially in patients of PTSD and severe anxiety. And this really opened up a resurgence of interest with the idea that there will be a legal status change eventually. A variety of new trials beginning to open up. Obviously, the work that happened with ketamine as a breakthrough drug that can be used as a rapid treatment for patients who do not respond to any other major therapy for their treatment-resistant depression opened up a variety of interesting ideas. And this gives you a sense of the interest in compounds of this class for depression and anxiety, for substance abuse, for pain, for sleep, for trauma, identity disorder, etc. And at sort of this time point, which is 2021, March, there are about 285 soon to be active and completed psychedelic trials that are being done all over the world to test whether some of these compounds, which originally really got pushed back because of the concern of them being, obviously they have concerns still, those concerns are not gone, but the fact of the matter is that there might be some breakthrough understandings for a variety of psychiatric disorders and that can come from these class of compounds. Okay, so I'm now going to tell you about what I do in my laboratory in the Department of Biological Sciences at TIFR in Mumbai. We have been deeply interested in serotonergic psychedelics for a long time. In fact, starting as early as my PhD thesis, I have been working on this particular molecule, DOI, which is a psychedelic, serotonergic psychedelic, prototypical serotonergic psychedelic synthesized that hits the 5-HT2A receptor. It's an agonist, which means it stimulates this receptor in the brain. And we approach, really what we're interested in asking is, we are interested in asking what are the effects that these drugs have on the brain, to try to understand them at a deeper molecular, cellular, and circuit level so that we can begin to explain how does the behavior change in response to these drugs. And I'm going to give you a sense of the kind of experiments my lab does to sort of start to address that. So a lot of the data I'm going to talk about today is actually unpublished. It's data that's happened in the last few years, and I thought I would share that. This is an opportunity for me to share that data with you. We're interested in how psychedelics produce hallucinations. This is perhaps why they were the biggest concern from the perspective of drug abuse. They do produce very robust hallucinations. We're interested in why they produce them. What are the molecular reasons that a hallucinatory state may be evoked by these serotonergic psychedelics? They also reduce anxiety quite dramatically, and we are interested in how. How is it that these compounds can reduce anxiety so effectively even in animal models, and how does one begin to study that and study the circuits in the brain that actually are responsible for showing a reduction in anxiety? We're interested in how they influence bioenergetics on the brain, and we stumbled on this a few years ago. Now, in 2019, we published a paper about this, which strongly indicates that psychedelics target mitochondria in the brain and can influence bioenergetics in the brain directly. And then we are also interested in how psychedelics in the brain may bring about a change in neuronal structure, like actually reorganize how neurons look in their shape, in their form. So I will walk you through this. I realize that the audience will be diverse and that there might be a little bit of jargon. I'll try my best to avoid jargon and to walk you through the story. So let's start with the idea, the first idea. So how does one even begin to study hallucinations in a rat whom you can't ask the question, are you hallucinating, right? They're not an available option for us. So if we are working with rats and mice, how do we start even looking at something that is indicative of a hallucinatory state? It's interesting that all the drugs that produce hallucinations in humans very robustly also evoke a very clear-cut response in rodents called a head twitch response. This head twitch response essentially looks like everybody will have seen this kind of a response, because if you have a dog anywhere, and we have lots of stray dogs running around, if you've seen a dog where water has fallen on its head or body, you know what a dog does, right? It shakes its head and all the water sort of falls off its head. So it's a very specific kind of, it's also called a wet dog shake or a head twitch response, where the animal just shakes its head almost like it's trying to get rid of water if the water is stuck on its fur. And this response somehow drastically goes up with every hallucinogen, but does not go up with non-hallucinogens. So here are examples from a paper from Gonzalez Maeso published in Science in 2007. Here are different synthetic compounds, DOI, DOM, DOB. All of these are synthetic serotonergic psychedelics. All of them increase head twitch responses. Here's mescaline, here is LSD, here's psilocybin. Here are non-hallucinogenic compounds which don't do it. And we know it's mediated through the 2A receptor, because this minus indicates mice, which are knockout mice, that do not have this receptor. Don't have the receptor, don't get the response. So we know that it's mediated via this receptor. I will show you an example of this so that you understand what do we mean when we say head twitch response. Okay, so here's a video. Please let me know if you can see the video. Did you see that little head twitch? It's a very specific.
Yes, madam, yes.
Yeah, I'll show it to you again, and you'll be able to see there's a very specific behavior that will show up. Yeah, that's the twitch. Okay, it's a very clear-cut twitch. It's very obvious. You can't miss it. And these animals show multiple such twitches if you look at it during a 20-minute window. So here is work from Toshali, a graduate student in the lab, and Shayasta, a postdoctoral fellow, where they've just quantitated the number of head twitches produced when the animal has had just an injection with like saline, because that is what we mix up the drug in. So here is DOI, here's a hallucinogenic compound. In 30 minutes, you can see these 20 head twitches. And in N-bomb, you will again get, N-bomb is again another synthetic serotonergic psychedelic. So what you're seeing is a behavior which is a hallmark of all hallucinogenic serotonergic hallucinogens are not seen with non-hallucinogenic compounds. So we use this as a proxy measure of the fact that it is producing a hallucinatory-like state. What we are also interested in is what is actually happening in the brain. So here is DOI, which is a molecule that actually goes and binds to this receptor, the 5-HT2A receptor, which is a G protein coupled receptor. And okay, there's a lot of jargon here, but these are all enzymes that are downstream, eventually resulting in an increase in calcium. And what we find is that when you stimulate this receptor with a drug that produces hallucinations versus a drug that does not produce hallucinations, you get a very different signature of all of these phosphorylated compounds downstream, either intermediates like IP3 and diacylglycerol, phosphorylated phospho-PLC, phosphorylation signatures of all of these different kinases downstream. And that already tells you high is red. So you can see that you get very strong response to the hallucinogen. You get somewhat of a response to a non-hallucinogenic, but it's not in anything like the league of what you see with these hallucinogenic compounds. So already that began to tell us that when you are stimulating this receptor with this drug, you're getting a very different signaling state than when you stimulate this receptor with a regular compound. Okay, so there's something unique that happens with these molecules. Then we were interested in asking, okay, so what is produced? And we were interested in the lipid molecules that are produced. And this is a collaboration we've done with Siddesh Kamath at IISA Pune, where he has done mass spec for lipid signaling signatures. And I'll walk you through this data. It will look like red and blue and pink or whatever. The more red something is, the more it is produced. Okay. So let's look at the first image. It says in vitro. What we have done is we have taken neurons from a rat brain. We've grown them in culture. So we have lots and lots of neurons growing in a dish. In the dish, we have either put DOI, DMSO, which means either it's just DMSO, which is just the control, or it is this compound DOI, and we are comparing it with lyseride. Now, DOI to lyseride, so if something is regulated, it will show up very strongly red. That means it is enhanced, okay? Lyseride is a compound that does not produce hallucinations. DOI is a compound that produces very strong hallucinations. So when you start comparing these, we are interested in seeing, can we separate out the compounds that are produced when something is producing hallucinations versus when it's not. And first, we start in a very simple system, not the brain, just neurons in a dish. So when we start with neurons in a dish, either we look directly at the neuron or we look at the medium in which the neuron is bathed. So we are asking, is something secreted? We're not only asking, is something made inside the neuron, but is something secreted by the neuron? And what you can start seeing is a signature pattern, which is quite different for the hallucinogens from the non-hallucinogens. So I'll show you something which is fairly in common. So at the bottom out here is these compounds here, which are fairly similar. DAG is higher here. There's lysophosphatidic acid, which is reasonably similar. But what you will start seeing is arachidonic acid here. And this is 2-arachidonyl glycerol. Okay, these are these compounds that we started getting very interested in because they are not produced when you actually stimulate the receptor strongly. You're still stimulating the receptor, but you're stimulating with a non-hallucinogen. You don't get those compounds. When you stimulate with a hallucinogen, you clearly get these compounds. So what are these compounds and why are they so interesting to us? First, we want to make sure this still happens in the brain because this is all in a dish, in neurons in a dish. So now we give it to a whole animal. We inject multiple animals with it. So we have lots of control animals which got saline, lots of animals that got DOI, lots of animals that got lyseride. And then we've taken out the brain, we hemisphereize, and then it goes to Pune. And then Siddesh runs a mass spec. And when we do that, again, we see, look at 2-arachidonyl glycerol. It's showing up as a hallmark signature, both in vitro in a dish and directly in the brain, telling us that when this animal consumes this hallucinogen, this product, this particular lipid signaling molecule is produced in large amounts. So we are interested in why this, what does this even mean? Okay. Now, why is this molecule interesting? It turns out it is an endogenous cannabinoid. Okay. Now I have to explain what an endogenous cannabinoid is. All of us feel pleasure. All of us feel a sense of joy. All of us feel a sense of happiness. Sometimes there is something that makes us particularly feel pleasure, right? Well, you have endogenous cannabinoids in your body which stimulate these cannabinoid receptors, and they drive some of our senses of pleasure, pleasure perception, all of that, right? Turns out that the receptors that are being stimulated are the cannabinoid receptors, and the molecule that is made by your body is 2-arachidonyl glycerol. Also anandamide, I won't get into that. And anandamide was not changed in our studies, but arachidonyl glycerol was drastically increased by drugs that produce hallucinations. And that's fascinating to us. And here's the biosynthetic pathway. We won't get into this. Why is this interesting? I will tell you what is the external molecule that stimulates this receptor, and you will begin to see why this is interesting. The external molecule that stimulates this receptor is marijuana. Okay, tetrahydrocannabinol is the active component of marijuana. Marijuana is also known as weed. Marijuana stimulates this particular receptor. That's why marijuana gives you the associated high. But obviously you make these compounds in your own body also, right? Because there is a reason you have the receptors. The receptors are not there in your brain to be stimulated by drugs that you consume. They're there in your body because they are regulated by endogenous or molecules you produce. One of the molecules you produce is 2-arachidonic glycerol. And it turns out that that's what is drastically being recruited by these hallucinogenic compounds. This is again just more evidence of it, showing that when you look at multiple different hallucinatory compounds, so here's DOI, TCB2, NBOM, all of them give you this increase in 2AG. Okay. The next question one can ask is it going through the 5-HT2A receptor? So we do hardcore pharmacology. What we do is we treat animals. So we have vehicle, regular animals, then the animals that were given the serotonergic psychedelic. We don't allow the drug to work on the 5-HT2A receptor. We simply block it. So when we block it, we block the HeadTwitch response. So the hallucinatory activity is gone. And what I want you to see is that the production of 2-arachidonic glycerol is also drastically reduced if you do not allow the drug to stimulate the 2A receptor. So that tells us that the 2A receptor is critical. There are other ways in which we can show this. We can use not just a pharmacological strategy, we can use a genetic strategy. So these are mice in which the 5-HT2A receptor is knocked out. It has been removed, so genetically deleted. So we have wild type, regular, healthy mice, which obviously have the 2A receptor. And we have 2A knockout mice in which this receptor is gone. When you don't have the receptor, and if you give the drug DOI, you will not get any effect. Obviously, as expected, there is no receptor for it to work on. And much the same way, the ability to produce 2-arachidonic glycerol is completely attenuated. So where does one go with these kinds of studies, right? So what we are very interested in asking is it possible that the reason why these compounds, these hallucinatory compounds, are producing such profound mood effects or effects on hallucinatory states and other states is because they're actually increasing your own body's production eventually of endocannabinoids by stimulating the 2A receptor. And what we find is that at least as far as the HeadTwitch response is concerned, this HeadTwitch response in animals is lost if you actually block the cannabinoid signaling pathway. And that's really interesting data that begins to tell us that, yes, it is a critical component. But what I have told you so far is really about what happens at the whole brain level. We don't know about which class of neurons. We don't know the details of this. So obviously what we are in the midst of doing is asking exactly that. We will now systematically go down to say which neurons, which class of neurons does it? How quickly does this happen? Where all does it happen? Which parts of the brain does it happen? So obviously there's much to be done. And what I hope I have given you a hint about is that we have stumbled on a very interesting finding that here you have this endogenous cannabinoid which your body makes, which is sort of hijacked by these molecules, and the production of this molecule goes up. That may be able to explain why some of the mood related effects happen. And obviously we have to study that way more extensively before we can conclude. We're also asking, does this happen in humans? Because everything I've shown you is rats and mice. So how do we do this? We can't do this in human volunteers, but what we can do is we are collaborating with people from NIMHANS, the faculty at NIMHANS, where they have taken fibroblast cells from human control patients as well as patients who have schizophrenia or depression. And you can grow neurons from these fibroblasts by using an induced pluripotent stem cell based approach. And here are examples of neurons that have been grown. They express the 5-HT2A receptor. And what we will ask is what we see in animal models, both in rats and mice, does it hold true for humans? And if it holds true for humans, is it different between healthy human neurons and human neurons derived from patients who have treatment-resistant depression or who have schizophrenia? So that's the angle. So let me switch gears now and tell you a little bit about our work trying to understand mood modulation, but from the perspective of anxiety. Okay. So how does one even begin to study whether these compounds regulate anxiety in animal models? So we have done this in rats, which are our favorite model organism. And I will show you some of this and I'll explain the data too. So this is the same compound DOI that we are using. What we do, and I'm showing you a sample test in particular. I'm not showing you all the data, but this gives you a sense of how we study this. Okay. So we have So animals are taken, large cohorts. There will be 10 animals that are given just regular vehicle treatment, 10 animals to 15 animals that are given the compound DOI. And within 15 to 20 minutes after they have been given these, they actually go into a behavioral test. So the behavioral test is done in a separate room facility where there are a variety of mazes. These mazes, and then the animal, it's completely screened off. So there's nobody watching, but there's an infrared camera so we can track the movements of the animal. So here, for example, is a tracking. When you see a lot of red, that's where the animal has spent most of its time over a 10-minute window. So this is just a heat map, but we can generate tracing maps. We know the speed at which they move, how much they move, where they go, how much time they spend, where they spend, whether they rear, whether they're sitting, because all of this is recorded, and we have automated tracking software that can track all of this, from their ambulation to their speed to their movement to all of these patterns of behavior. And what are we looking at? So this is the maze. The maze is an elevated plus maze in which there are two open arms which are raised about 2 feet off the ground. So here the animal can fall off, right? So it's a little bit of a scary space for the animal. And then there are two arms which are completely closed off with high walls. So in general, the animal prefers to spend time in the closed arms. Okay. So I'll show you what the video looks like just to give you a sense. So this is just a cartoon. So the animal will go spend time in the closed arm. Every now and then it will go into the open arms, check it out and come back. And it's doing this, and we are tracking the behavior of large numbers of animals. And what we saw remarkably, and that's obvious from the heat map here, the heat map shows the way an example of a particular male rat which has been given vehicle control. Here's an animal that was given DOI. The animals that are given DOI like to spend their time in the open arms, which means that their anxiety is drastically reduced. Normally, you will not see animals going and spending this kind of time in the open arm, because the open arm is the part of the arm which is the most likely to evoke a sense of fear, because there's a risk it's completely open and you could potentially fall off. Also, you're very exposed. So, which the best way I can describe this is, anyone who has seen a rat scurrying will realize that it likes to hang on to the corners. It doesn't run straight across the room. Even if you have a mouse or rat in your house, it will cling to the wall and hide like that and hide away, right? It's not going to run straight across the middle of the room. So much the same way, even in our animal house, these animals, when they're put in these mazes, they prefer to sit in hidden corners or hide away. And so this kind of behavior is rather unusual. But when they take this drug, the serotonergic psychedelic, they spend much more time in the open arms and less time in the closed arms. This is true whether you're dealing with a male rat or a female rat, because even female rats given the same compound will show a very similar phenotype. Okay, so they also show a reduction in anxiety. They spend much more time in the open arms. And this is also true in a completely different species, in mice. Mice also prefer to spend time much more in the far less time in the closed arms and much more time in the open arms. All right. So what do we do with this information? What have we done? We have replicated what is known to happen in humans. In humans, if they are given serotonergic psychedelics, anxiety is reduced. This has been seen with LSD. This has been seen with psilocybin. This has been seen with MDMA. So it is known from clinical data. In fact, there are trials underway to potentially use very low doses of these compounds to actually reduce anxiety. But how do you study this? Now, how do we even figure out which part of the brain this drug is working on? So to figure that out, we have to deliver the drug instead of giving it to the whole animal, which is where it was given. So you either give it per os, which is either the animal eats it, or you give it intraperitoneally into the peritoneal cavity, and then that it's transported everywhere. And you don't know where it's worked, but it has given you a behavioral effect. So how do you start dissecting this? So one way in which you can dissect it is to deliver it to specific parts of the brain. So to do that, you have to stereotactically deliver the drug through cannulae that are actually placed into individual parts of the brain, and then the drug, so that first the surgery is done. We do the surgery on 3 to 4 month old rats, and here's an example how it's a cartoon animation. I'll show you how a real surgery is done. So the animals go through surgery, they're fully anesthetized, the surgery happens, and they have 2 to 3 weeks to completely recover. And we place cannula in the brain. And so here, for example, is a drilling. And once, so suppose you are drilling 2 holes here, then you will actually implant cannula, shown you here in this gray, so that now if you want to deliver through this stylet, the drug will be delivered straight to only this part of the brain, right? So these are the fairly complex stereotactic surgeries, and we want to now ask which part of the brain is important. The problem is there are lots of parts of the brain that regulate anxiety. And I'm just showing you some examples. These areas of the brain and the rat brain right at the front, these are the prefrontal cortices, the prelimbic and the infralimbic, they also regulate anxiety. The amygdala also regulates anxiety. The ventral hippocampus and the dorsal hippocampus also regulate anxiety. This is just a half picture. There's the bed nucleus of the stria terminalis. There's the hypothalamus. Where do you start? So a very talented graduate student in my lab, Prachi, decided she was going to go after three main regions: the prefrontal cortex, the amygdala, and the hippocampus. And she's done more than 1,500 surgeries in the last few years to start nailing down which part of the brain is absolutely responsible for this particular behavior. And here is what she has shown, and I'll show you her data. So this is work from Prachi, senior graduate student in the lab, assisted by another graduate student, Darshana, Amartya, master student, and Sarvesh, who is a first-year graduate student. And so here's how the surgeries are done. You'll be able to see them on the side. The animal goes into an anesthesia unit. There's complete anesthesia here. Then you cut through, you open it up, you dissect, and you actually start implanting. And then you deliver cannulae into different parts of the brain. And what I will tell you is she did surgeries for many areas. Here is the data that showed us it was the ventral hippocampus. Because when you place the cannulae in the ventral hippocampus and deliver drug only into the ventral hippocampus, you reproduce what happens when the whole animal takes it, right? Ingests it. So you're producing the same kind of a phenotype, telling us that the effects are mediated through the ventral hippocampus. And we looked at multiple other regions. So here is the dorsal hippocampus, no effect in the dorsal hippocampus. Here is the amygdala, no effect in the amygdala. I'm not showing you data for the PFC, but both the IL and the PL structures, they're also no effect. So 5 regions looked at. Out of 5 regions, there's only one part which clearly gives us an effect. So now that's very interesting, because what have we done? From saying that these drugs actually reduce anxiety, we are now able to say with confidence that the reason they reduce anxiety is because they actually work on the ventral hippocampus in the brain. But that's not enough, because there are millions of neurons in the ventral hippocampus. So which class of neurons? How does it work? Obviously, that's the next question one would want to ask. So the next question we want to ask is, we want to absolutely be sure that's where it's working. So the experiment that Prachi did is she cannulated and she blocked the serotonin 2 receptor only in the ventral hippocampus. So first the cannulation is done, then the animals are allowed to recover. Then you put in the drug that blocks the serotonin 2A receptor here, and then you actually inject the whole animal with the serotonergic psychedelic. So your hope is, if this receptor is very important in this region, then even though you've given the drug to the animal, it should not work, because you've blocked the receptor in the correct part of the brain. And that's what she sees here. So here you can see this is DOI. Here is DOI in vehicle. Here clearly reduces anxiety. Animals are in the open arms, far less in the closed arms. But when you give this drug to animals where you have blocked the receptor only in the ventral hippocampus, the drug doesn't work. So now this is one more proof of the fact that this region of the brain is where the effects are being mediated. Because even though you've given the drug to the whole animal, and that is given intraperitoneally, if you block the receptor in this part, you completely block the effects. So where is it working now? Which class of neurons in this part of the brain? Turns out the serotonin 2A receptor is expressed by a class of neurons here called the paravalbumin neurons, the PV neurons. Okay, so it's working on this class of neurons, and I'm going to cut through some of this quickly because it's a fair bit of data, but okay, let me just quickly describe it. What we have is actually wild type animals here. So these are regular animals that were given, they have the 2A receptor. It's not taken out, okay? So when you have the 2A receptor, you should expect it to work. So if you now deliver DOI straight into the ventral hippocampus, you will get a reduction in anxiety. This is indeed what you see in the elevated plasmase. Then we have a mouse in which the receptor is removed, completely removed. So when it is completely removed, you lose the effects because there is no receptor for it to work on. So clearly you need the 2A receptor. That much is clear. This is a collaboration with Noelia Weisstraub and Jay Gingrich at Columbia University, who send us these knockout mice, and we breed them at TIFR, and then we are able to do these experiments, but they generated the mice. So they are collaborators for this project. Now what we want to do is we want to put, so we have a wild type which has 2A receptor. We have a knockout in which there is no 2A receptor. Now we want to selectively put back the 2A receptor only in the paravalvumin neurons, only in the ventral hippocampus. That's the goal, right? So we attempt to do that using a rescue-based strategy. I won't get into this, but there is a cool genetic tool that allows us to actually put back the receptor in an animal that does not have the receptor, only in a subclass of neurons. And that's just genetic tools that we can use to actually do this. And when we do that, we rescue back the behavior. And so that tells us that if this animal has absolutely no 2A receptor, but the 2A receptor has come back in the ventral hippocampal PV neurons, then we can get an effect on anxiety. And that really opens up the question of this circuit. This is the circuit that it's working on. Where are this class of neurons projecting? If you inhibit this circuit, because that's what these receptors would do in this particular neurons, they would actually activate PV neurons, which would inhibit the ventral hippocampus. How is this working? So then the natural question to ask is, what if we just inhibit the ventral hippocampus? Forget these drugs. We won't do anything with the drugs. We'll just use a nice genetic pharmacological tool technology and we will just switch off the ventral hippocampus. If we do that, will it produce the same thing that these drugs do? And so we inhibit the ventral hippocampus, and sure enough, we produce these effects, not as strongly as the drugs, but we are moving in that direction. We can clearly see that if you inhibit the ventral hippocampus, you get a similar effect. So then we ask, which projection is it? From where does it go to where? And we have a way of tracing the pathway from the ventral hippocampus to the amygdala. And when we do that and we inhibit this pathway, then we are actually able to reproduce everything that the drugs do. So there's no drugs in these animals. I mean, there's no psychedelic in these animals. We've just perturbed the pathway we think that the psychedelics are working on. We use the psychedelics to understand the pathway. We can perturb the pathway. And when we perturb the pathway, we reproduce the behavior. So that strongly tells us that what we believe is happening is serotonergic psychedelics are working on the ventral hippocampus to inhibit the ventral hippocampus. By mediating this, they actually reduce anxiety and avoidance behavior. We are in the midst of a big collaboration with Alex Kwan at Yale and Rishikesh Narayanan at IISc to actually record from neurons while these animals actually have the serotonergic psychedelics on board. And hopefully we will take that idea and take it further. How am I doing for time? Do I have a few more minutes?
Yes, you can continue, no problem.
All right. Yeah. So I then now like to end by talking about our work on psychedelics and mitochondria, which we just completely stumbled on. So my lab is much more a behavioral lab, a lab that looks at circuits and looks at molecular regulation of the circuitry to produce particular behaviors. But we're also interested in what might be the cellular consequences of some of these compounds. And what I'm going to show you is data that indicates that serotonergic psychedelics may directly impact mitochondria in neurons. So neurons, when grown in vitro, here is a culture. Here are beautiful MAP2 positive neurons taken from the cortex of a rat. They express the 5-HT2A receptor because it's abundantly present. And it's expressed here is just mRNA expression showing that there is the receptors in the right neurons. What we find is that when we stimulate these neurons with these serotonergic psychedelics, the amount of production of ATP, which is the energy currency of neurons, or all cells actually. So the energy currency, which is produced by mitochondria, but also produced through the glycolytic pathway, this goes up, and actually the number of mitochondria, so the mitochondrial mass in a neuron also goes up. So this is work that's done in collaboration with Ullas Koltur at TIFR and actually with my father as well, Ashok Vaidya at KHS. And this is work done by a very talented postdoctoral fellow who's now an assistant professor at KHS, Sashena Fanivanda. So she then went on to ask, is there actually a change in the oxygen consumption rate, which is the utilization of oxygen by cells to eventually produce energy? This is how all our cells respire, right? They take oxygen, they use it to produce energy. That's how cells do all of their activities. The energy currency being ATP. Turns out when you have the serotonergic psychedelics on board, you do more oxygen consumption. So your basal respiration goes up, ATP coupled respiration goes up, maximal respiration goes up. Essentially, you are using more oxygen and more respiration to actually produce more energy equivalents for the cell. So the cell has sort of ramped up its bioenergetics when it's exposed to a serotonergic psychedelic. And this was very, very interesting because that's seen not only in vitro in culture. If you treat rats with these compounds for 3 days and you actually look at the mass of mitochondria, that goes up. There are other measures that confirm that, many other molecules, both at the mRNA and protein level, that show us an increase in production, which is indicative of more mitochondrial production. So increase in ATP levels. And when you isolate mitochondria from the cortex of these animals that have received serotonergic psychedelics and then look at the oxygen consumption rate, The OCR is up, telling us that respiratory capacity and respiration is up. And that's quite fascinating because it tells us you're ramping up bioenergetics in response to these drugs. This is not seen only with this compound, it is seen with other synthetic serotonergic psychedelics like TCB2 and NBOM. But fascinatingly, we've started an interesting collaboration with my father, Dr. Ashok Vaidya at KHS, where there's a deep interest in looking at compounds derived from traditional Indian medicine. Because it's not as though these were compounds that were used only, like ayahuasca was used by the shamans in South America. There have been reports of hallucinogenic compounds used in traditional Indian medicine also for centuries. But what do those molecules do? What are the molecules? How are they working? What are they doing? We don't really know. So we were very interested in following up with some compounds which have these kinds of reported effects in Indian traditional medicine. And so we decided to ask, if we just take one of them and use them, will we also get these kind of mitochondrial effects? And what happens if you don't have the serotonin 2A receptor? So here is berberine. It's an isoquinoline alkaloid. And here is berberine, which is the active component of Berberis vulgaris. And when you use this compound, it is quite fascinating. It gives you robust effects on ATP generation. But if you don't have the serotonin 2A receptor, nothing will happen. So now we have a molecular target. For this particular effect of berberine, we know it requires a 5-HT2A receptor, and that starts opening up a whole new idea of potentially looking at plant-derived indole and isoquinoline alkaloids that come from Indian traditional medicine that may also potentially have these kinds of effects, both at the molecular and cellular level, but also at the behavioral level. And we're very interested in pursuing that as well. This is just to show that we have now figured out some of the pathways that are downstream. I told you that the serotonin 2A receptor is critical. We were able to show that two molecular mediators, SERT1, which is deacylase, as well as PGC1 alpha, which is a transcriptional coactivator. These are central to these effects, because when you start looking at some of these effects, when you knock out PGC1 alpha, here's SERT1 knockout, you lose the effects on mitochondrial DNA content and you lose the effects on ATP, telling us that these pathways are essential to how these drugs and compounds are actually working. But what does it mean? So suppose you increase energetics. What's the great value of increasing energetics? I mean, is there some good functional consequence of increasing energetics like this? Well, it turns out something fascinating happens when you do that. When you do that, you protect the neurons from oxidative and excitotoxic stress quite drastically. So here you have DOI. So minus will be there's no DOI. You're looking at how many cells are surviving. So percentage cell viability, 100 is what we have normalized everything to. That's our control. When you apply DOI, it doesn't do anything by itself. Here's when you come in with an excitotoxin like kainate. Here's when you come in with H2O2, which is an oxidative stress. What you can see is a clear reduction in survival. Comes down to close to 50%. So half the cells die at this particular concentration. Not surprising because these are very strong toxins. But if you come in with DOI, look at what happens. You rescue. So even at higher concentrations, you're still rescuing. And that's telling you that when you have had exposure to these serotonergic psychedelics, these neurons are buffering excitotoxic stress, they're buffering oxidative stress. And that's also obvious here when you look at it with reference to a dose response curve. So here is kainate in blue, cells die as you increase the concentration. If they've been exposed to the serotonergic psychedelic DOI, they are protected. You can see that here also. But if you do not allow the SERT1 molecule, the catalytic activity of the SERT1 molecule, you lose all these protective effects. So that tells us SERT1 matters. So collectively, these things are opening up a lot of new ideas. These are molecules that have been used for thousands of years, but we have a poor understanding still of what do they exactly do in the brain. Yes, they produce hallucinations, which is the concern and worry with these compounds, because they are, in that sense, they can become risk factors for certain psychiatric disorders, in particular schizophrenia or a psychotic episode. But at low doses, they have profoundly beneficial effects on both anxiety and mood. They are robust regulators of bioenergetics and they're strongly neuroprotective. In the interest of time, I'm not going to tell you about our work with psychedelics and neuronal plasticity, directly addressing how they change shape of neurons and what are the molecules that mediate that. Instead, I'm just going to quickly wrap up by acknowledging the very large and talented teams of people that do this work. So the work on serotonergic psychedelics, hallucinogens versus non-hallucinogens, looking at the lipid signaling signatures is led by Toshali and a lot of other people in the laboratory. My collaborators here include Siddesh Kamath, Ritika, Likhansh, and Viju at Nimhans. Our work with anxiety-like behavior is led by Prachi in the lab and a bunch of other people. Our collaborators are Rishikesh Narayan and Alex Kwan. Serotonergic Psychedelics and Bioenergetics, led by Sashena and Asma. My collaborators Ashok Vaidya and Ullas Koltur. And we're very interested in opening up the idea that traditional Indian medicine leads similar to what were used by the shamans and were used in Africa. The psychedelics and plasticity work I didn't get a chance to talk about today. The funding comes from TIFR DAE as well as from Sri Ramakrishna grant from the Padmavati Ishwara Foundation and from India Alliance Fellowship, two of my very talented postdoctoral fellows. And this is my team of people. There is a reason we have a psychedelic mushroom. Everybody is showing it because that's what we work on quite extensively. Two very talented people who have now left the lab but who started some of this work and were seminal in getting it off the ground. My collaborators who have been invaluable in our being able to pursue this. And I am done. Very happy to take questions. Thank you for your patience.
Yeah, thank you, Professor Vidita. It was a wonderful lecture. Only I had a lot of hitches in the beginning. I couldn't join. I see you're so passionate about your subject, and I like the word that mitochondria. Of course, it is mitochondria, and it affects our moods. I would start off with a few questions, and then I'll ask. You have told so many things about the drugs and all, but basically we can just talk even about coffee being a stimulant, or even an ice water being a stimulant. Can you just tell us as a layman how it works? Because we are not into this kind of subject as such.
Caffeine will not give you this class of effects. It hits a completely different class of neurons. So it's not going to fall into this sort of a category. But caffeine is also dependence inducing. Those of you who drink coffee, if you miss your nice strong filter coffee in the morning, have a mild headache and feel completely out of it. The reason you feel out of it is because you're getting a caffeine withdrawal, and you need that boost of caffeine just to give you the feeling of being completely okay. But yes, it is a stimulant. Yes, it is also dependence producing because it works on a different population of neurons. Iced water, perhaps not so likely to do anything other than make you feel good. Depending on the Mumbai heat and weather, it might make you feel good. But caffeine, absolutely, because caffeine is a potent molecule. So is sugar, by the way. I mean, sugar is equally a strong drug of abuse. It's just that we put it everywhere. That's all. It's a dependence-inducing drug as well. Sugar can produce a lot of dependence as well.
Yeah, now I would throw the talk to the audience. They can ask any questions, or they can put it in the chat box in YouTube also. I could read it out.
Dr. Vaidya, may I ask a question?
Absolutely.
First of all, I want to thank you for a very, very fascinating lecture, going deep into the brain and to the molecular level to understand how anxiety can be caused or no, treated. I I was just curiously watching your talk with regard to your own terminology and I found that you are using a word fascinating almost 3-4 times every minute. So that shows how nice is the subject. Now let me come back to this question. Finally, what does it mean for treating patients who are having hallucinations? Where does it go from here?
Yeah. So where it is going from here for the psychedelic compounds is that low-dose psilocybin is now potentially being thought about as a way for assisted therapy for patients who have severe post-traumatic stress disorder and are not responding to most of the other sort of milder classes of drugs. It's also being considered for severe anxiety and is being considered for treatment-resistant depression. And so these trials have rolled out. It is already being used in the United States, not actively yet being used in India as much. Ketamine is being used, but I see it as a matter of time. The worry, of course, as always, with using these compounds is if the individual has a prior potential history of schizophrenia or a risk for schizophrenia-like behavior, then the hallucinatory component of these compounds is worrisome, obviously. They're not toxic compounds, but the fact that they can produce hallucinations is a worry. And so they are trying to use, how do you do this without actually, so there are lots of interesting molecules in the pipeline which do not produce hallucinations but can mimic these effects on anxiety and mood. So, there's a compound which is derived from the iboga plant, which is called tabernacle, used by David Olson's lab, does not produce hallucinations but replicates much of the other positive effects. So I think what you will start seeing is a synthetic chemists, combinatorial chemistry kicking in big time to see if we can come up with ways via which we can produce one component but not the other. And that is absolutely possible because there are plenty of compounds that hit these receptors without producing hallucinations. So it's just a matter of tweaking the chemistry to get that right, to be able to produce signaling signatures that give you the beneficial effects without giving you the negative effects. So I see that as a revolution in the making in the next 20 years. Yeah.
Okay, just attendant question is, instead of delivering a drug to a person and influencing his behavior, you mentioned that at the neuronic level, if certain pathways are blocked, these chemicals do not cause hallucinations. Yeah. So for a hallucination patient, is it not possible for you to do that rather than give a drug?
So for a hallucinating patient, the drug that is used is actually one of the drugs that works really well is a drug called clozapine. And clozapine actually blocks the HT2A receptor, which makes complete sense. I mean, clozapine is what is used for patients of schizophrenia quite routinely. Unfortunately, it has side effects because the problem with all of this is the 2A receptor is not only in the brain, it's everywhere else also. So when you block the 2A receptor, you will also get some of these peripheral effects that start being a bit worrisome. But people use the blockers of the 5-HT2A receptor in any case for treating psychosis, for treating schizophrenia also, quite extensively. What becomes interesting is what can you do to activate your own endogenous endocannabinoids? What I showed at the very beginning, which is, it's not like you don't have the ability to make your own endogenous endocannabinoids. Obviously, the drugs make large amount, but exercise becomes extremely interesting. So does enriched environment. So does sort of stimulating the nervous system through certain tasks that might actually drive an increase in endogenous production of endocannabinoids, which could then be used without a drug-mediated interventional approach. And that is also something that's well worth considering and thinking about.
Well, my last question would be that you mentioned even about your ability to locate a specific region inside the brain which is the center point of action, and perhaps other regions are not very important. So instead of delivering a drug, is it not possible now to think of microsurgery at that level and maybe introduce something which can continuously deliver a drug for a long period of time on a sustained basis or whatever? I'm just thinking, are there other ways of doing it instead of making a patient rely on regular, every day you have to take a drug? If you miss it, you are in trouble. Can you not avoid that?
I wish there was. I mean, I don't think the only real invasive surgeries into the brain run their big risks, and that's why that has hardly been an approach unless you consider Parkinson's disease, where it's done, pallidotomies are done just because patients who are no longer responding to any drug, then they do surgery of that kind. It's not an easy area in which to do surgery, unlike delivering something to the liver, the kidney, the body, the skin, even systemically. Getting things into the brain is a tricky job. And because it's a highly vascularized structure, the risks of infection, the risk of, it's complex. And so I don't see that happening. What I see is more likely to happen is, are there ways to perhaps target subclasses of neurons using better and better design of tools and approaches. It will happen. I don't think it'll happen in the next 20 years. That is something that is going to happen over a period of time. I see people using a variety of approaches to see if you can actually stimulate neurons very selectively. I mean, there's transcranial magnetic stimulation. It's not done to stimulate such narrow areas, but it is used right now as therapy certainly for major depression. So you will see that beginning to emerge, but I don't see surgical intervention emerging as a major avenue, only because it is, see, with rats, we have a very high survival rate. Our survival rates are like 99.6%. And most of the animals we do surgery on, all of them survive. And that's because, first of all, rats produce their own vitamin C. So their likelihood of getting an infection is very low. Whereas the likelihood of human infection post such an invasive surgery tends to be high. And so I can't imagine down the road for anything unless it is intractable and doesn't respond to any drug that surgery will become an avenue.
Okay, there is one question in the chat box. Somebody has asked that with this type of research, can ADHD behavior in children your research be used for that?
That's an interesting question. People are talking about it from the perspective of autism spectrum disorders, perhaps not specifically ADHD. ADHD is often treated with drugs like Ritalin, which target the noradrenergic pathways, a very different pathway in the prefrontal cortex. But for autism spectrum disorders, because social behavior is enhanced with low doses or some of these compounds, they are in preclinical studies quite extensively in autism spectrum disorder models to see if they have a beneficial effect. So that's something that's in the pipeline. It's not, there are no trials on this whatsoever, but certainly at the preclinical level, there are lots of groups around the world that are beginning to look at that. ADHD less so, but certainly autism spectrum disorder.
So you mean to say autism can be treated by a specifically reaching some part of the brain?
So there is evidence that some of these compounds, some of these serotonergic psychedelics, they also reduce social anxiety and enhance social behavior. And one of the hallmark indicators with autism spectrum disorders is a complete social phobia and social anxiety, as well as social issues associated with that. So for that component, these molecules are being tested in currently animal models. I imagine that it will go from that to primate, etc. But certainly right now in animal models, they're being tested.
Is there anybody else who wants to ask a question? Can you please put this in the chat box? I'm seeing the YouTube also. I don't find any questions there.
So if you are waiting for some questions, maybe I can add another question, which may be rather maybe philosophical or silly. I do not know.
Yeah, please.
Usually, our discussion as scientists, chemists, biologists, whatever, we talk about molecules influencing the brain and behavior. Now, what do you say about music influencing? If there is no molecule there, and one can say that a person's psychological state can be affected by listening to a very good music and so on. How does music work on somebody's brain when there is no molecule?
But there are molecules. Absolutely, there are molecules. In response to phenomenally beautiful music, which you appreciate greatly, you will secrete endogenous endocannabinoids absolutely to produce a pleasure response. And so there absolutely is a molecular response in the brain. And even in rodents, people have done this where they have actually made animals listen to classical music versus extremely loud rock music, and you get completely different effects in the brain, because obviously volume will matter, but also tonality, nature of rhythm of music, etc., make a substantial impact on the nervous system. That is not it's a great question. There are a lot of people beginning to ask, where exactly in the brain do you see specific changes? And music as a tool and music as a therapy is also something that is quite interesting in its own right. So yeah, I mean, it's perturbation that absolutely has an effect on the brain. No question about it. Just like exercise does.
Okay, are there no more questions? If there are no more questions, I would like to just give a thanks to Professor Vaidya. I'm sorry I could not read out your biodata at that time, so probably now I can just tell. We are very happy to have a Shanti Swarup Bhatnagar awardee among us. I'm sure it would have been very encouraging for the students to listen to you. And of course, there are a lot of people who are working in biosciences in the HBNI, and then I'm sure they would have been benefited and inspired by you. So thanks a lot for being here today, because we were not able to schedule it earlier, and then finally we caught it on that. Also, I know that you are committed to mentorship and the equity and diversity in STEM, which is recognized, and you were recognized recently for a Nature Award mentorship in a mid-career category in 2019. Also, she's a recipient of the J.C. Bose Fellowship recently in 2021. Her research group is very much interested in understanding the neurocircuitry of emotion, its modulation by life experience, and the alterations of emotional neurocircuitry that underlie complex psychiatric disorders like depression. Of course, we heard a lot of it today, and then we are very glad that you're doing this kind of specific research in a specific topic, and you are having a great team. The great team is supporting you. Thanks a lot for giving us this lecture. We are benefited by you, and we'll contact you more also in future.
Thank you. Thank you. My pleasure. Thank you very much for patiently listening. Thank you. Bye-bye.
Bye-bye.